Yeah, so today I'm going to talk a little bit about both how to start exploring data. If you have collected a data set in the lab, or if you've downloaded something from a paper, how you can start to explore it and visualize it in different ways. And then we'll continue with an introduction to descriptive statistics. And then the next two lectures, so this will sort of be a bridge between the Python part of the class and the rest. And after this, there are two lectures that Stuart will give on sequence alignment and sequence database searching. And then there will be a statistics block of 5 lectures. And this is sort of in between the programming and the statistics a little bit, this lecture. The idea with this lecture is that you will get used to a library in Python called matplotlib, and that you'll learn how to use it to visualize simple data sets, and how to make scatter plots, histograms, how to make a kernel density estimate, and box plots. So especially the box plots is something that would be really good if you got familiar with, because it's in general a much better way to visualize data than to use just bar graphs. Then we'll talk a little bit about descriptive statistics, about how to what mean and median means for different kinds of distributions, and also looking at different measures of variation, like standard deviation, interquartile range. And then we'll talk about the central limit theorem, and that will be also the homework. So if we start with a simple data set, so let's say you've measured something in the lab and you've collected these 20 data points, and this is a repeated measurement of the same underlying parameter, and it doesn't matter what it is, and we'll just it's a list of 20 numbers, and so the question is how do we approach this? How do we start looking at this to make sense of what this means? So if this now is meant to be listed in the order that the data points were obtained, so first thing we can do is just to plot them in the order that they were obtained. So here now we see that there is some variation, and the value seems to be somewhere close to 0, but varying around it. So the advantage of starting with a plot like this is that you can see if there are any systematic errors. If there are, for example, if the measurement changes over time, which is not unusual. Let's say you start with the first measurement in the morning, and then you continue measuring for the whole day, and the temperature in the room rises maybe a few degrees, so maybe your instrument changes the accuracy and what you measure. So it's very useful, but here if we look at this, there is no obvious trend that we see, so we can probably say that it looks reasonably okay, but in other cases we might see that the value just increases as we measure, and that's not so good. So, but we get some idea of what the spread is around the mean, but there are probably better ways. And one way to look at the spread is to plot a histogram. And so below there, so this is, again, it's the next few slides will be the same 20 data points, just plotted in different ways. So on top we have our the same plot as in the previous slide, but then we have made histograms here. And the one thing with when you make a histogram is you have to decide what the bin size should be. And so we see 3 different bin sizes, and probably this one, the bin size is too small, because we have too much variation between bins, so that's not a good choice of a bin size. This is probably a better choice, so somewhere between 0.1 and 0.01 would be reasonable for this data set of 20 points. But here now we can get a better idea of how much variation we have. Yeah, sorry. So a bin size, so you see these vertical lines. So when we make a histogram, we divide the x-axis, or in this case, we divide this axis here, how much we deviate from 0, into bins. So we decide that, so we have, so in this case, if we have 0.05, we have between 0 and 0.05. We count how many points there are. Then between 0.05 and 0.1, we count how many points there are. And then we plot on the y-axis here the number. So for example, in this bin here, so between 0 and 0.05, there are 7 of these dots fall in there. So that's how and then we do this over the whole range that we're interested in. And so is that, yeah. So the problem comes when you use histogram is that you have to choose how wide the bin should be, and then when you compare to histograms, it's not very convenient because they have these steps, and it's not. So we're going to look at some methods that are easier when we compare different distributions. So one thing we can do instead is to make a cumulative distribution. So now we start at -1 here, and then count how many points we have that are smaller than -1, which is we don't have any points there. So we have, we're 0. It's 0. So then we walk up, and then when we meet the first point, which I guess is this one, then we take a step up here. And then every time as we slide along here, when we meet the point, we go up. So then by the time we've reached maybe 0.2 or something like that, then we've counted all the points, so we've reached the frequency of 1. So we have all our data is between these 2 points. So now, as you can see, this is a smoother curve and than the histogram, and this will be much easier to then also compare when we have different data sets, as we'll come back to. So another way to get a smoother distribution is what's called the kernel density estimate. So it's what we do is we look on for each point of the measurement. What we have, the value we measure, we put a small smooth distribution at that point, and then we add them up. And here now, because of that, we have not a we don't bin it, but we add a small, usually a Gaussian distribution, and then around at each point. And that will add up to this distribution here. So now again, this here we also have to choose a width of this underlying Gaussian distribution, but the final distribution is less sensitive to that. and this is something we're also going to see how it compares when we compare different distributions. So now this is just the summary of this data. So now we had these 20 data points. And so first we just looked at them in the order they were measured to see if there were any systematic trends or systematic errors that were introduced. Then we looked at making histogram and the kernel density estimate of the same data. And now we can compare this to the original distribution, which is, in this case, just comes from a normal distribution with a mean of 0 and the standard deviation of 0.1. We can see that both these measurements sort of recapitulate that. The kernel density estimate is slightly better at getting closer to the original distribution. But the problem is that we only have 20 points, so we're still not going to be able to get back to the original distribution very well. But if we increase the number of points, so in this case now we have instead 200, then we see that both the histogram and the kernel density estimate gets closer to what the original distribution is. And of course, when we do measurements, what we want to do is to sample this original distribution as well as possible. So now if we go back to how these plots were created. So we'll start with, if you have downloaded this, the first Python script, you can see that what we did to get those 20 points was to draw 20 times from a normal distribution with a mean of 0 and standard deviation of 0.1. And that we can simply do by, first we need to import NumPy, and then we select a sample size of 20 using this random normal, and we multiply it by 0.1 so to get the standard deviation 0.1. So if you can try this and see if you get a list of 20. Have you all been able to download the script? Anyone who hasn't? Sorry?
Yeah, I'm saying.
You have? Okay, great. Yeah. I'll give you a minute if you need to.
How does it know the data that is importing it?
So this is not data. This is just random generation. So this is really a random sampling from the distribution. So every time you run this, you will get 20 different points.
Oh, okay.
And you will, so when you run these scripts, you will get slightly different, but it's still sampling the same distribution. So for example, so now if we do this, when I did it, I got these numbers.
How do you set mean is 0?
Sorry?
How do you set
Oh, so the mean, it's 0.
that's
yeah.
the default.
That's the default. So what you can do if you want to change the mean, you just add a number to the whole array. We'll do that later in a case. So yeah.
I was just going to say, so it's the code you're using that's normal is asking for normal distribution. Random would be random numbers that you're pulling from a normal distribution, and you're asking for 20 numbers.
Yes,
Okay.
exactly.
Okay.
And so that's what, yeah, so this was what I did, I mean, for the first figure that I showed. So that's, yeah, so this is what I get. You're going to get different numbers when you do it, but it should be sort of in the general range, same general range. So now to actually plot these, it's very simple with Matplotlib. And so first we have to import it in this first what we do here. And so you can just, these things you can copy from the scripts that you downloaded, so you don't need to type really anything, so you can just copy and paste. So then, because we decided we wanted to plot the points according to the order that we sampled them, we need here to define an array that goes from 1 to, in this case, 21. So the point is 20. So from 1 to 21. So that's how we plot it. And then we need to set up a figure. So we define this figure and an axis. So in this case, we have just one subplot, but you can have many. So, but in this, all these examples, we're just going to have one plot per figure, just to make it simple. And then we say that its size should be 6 by 6 inches. But that you can also change if you want to have that differently. So then the actual plotting is this, what's shown here in bold, is that on axis 1, we do a scatter plot. So, and the x, which is this array, which is goes from 1 to 21, 1 to 20, and then y is our 20 randomly sampled variables from the previous plot. And then we're going to color them red, and we don't want any line around the symbol, and then we want the size 40. And then the rest is just to set the x-axis limit, the y-axis limit, and save the figure, and then finally close it. So this is, so if we do this, you should get something like this, but with a different set of data. And does that work for you?
Do we have defined points? Like, I try to copy and paste.
Yeah, so I guess so you should, you should define points as 20. I should have changed that. In the script it should be fine. It's, you should have it, it should run.
Just say points equal
20, yeah. Yeah, so sometimes the thing what I copied here might miss some of the things, and but in the script it should all be defined, and because I just ran the scripts before, and it all produced the plots.
You want to put points equals 20, and then it's going to come out as a PNG file in your whatever folder you suggested.
Yeah, and it should save this PNG file in the same folder as your script is in.
Yeah, so you just put the above the x points equals 20, and then 1 plus 1 means 1 to 20.
Anyone who's running into?
Just a question, like when you generate the figure, where is it stored temporarily?
I mean, so until you save, it's in memory, in your program. But then when it reaches the save fig command, then it will save it. And in this case, it will save it to the same directory as your script is in.
So figure is just a random name for the figure. It's not.
Sorry,
So the FIG, the fig is.
Yeah, you can call it something else. And the X1 is also, you can call it anything you want. And we might later call it some other things. I'm not sure. I think I try to keep it the same name. But yeah, it's that you choose. And if you want to, if you have a complicated data set that you want to create many different figures, you might define several figures and then.
Yeah, wherever I save that's going to be the title, counterpoint plus string, and then that should be saving wherever wherever we print out the stuff for the last time.
Okay. Any? Have you? Everyone succeeded to create a figure? No? Almost. So well, I'll continue here. I mean, if you have problems on Thursday in the tutorial, you can go through. Yeah.
I was wondering what each of those, in the parentheses where you do the big.save big, what each of those things mean, like the DPI and the dots?
Yeah, so the DPI is dots per inch. So that's how, what quality you want the figure to be. So you can put it lower if you want to save space, for example. So DPI 300 is usually if you want to print it. If you just want to show it on a website, it might be enough with less. Yeah, you don't need to change it. And so also, and it's difficult to remember all these things. So the best is to just Google and see what other options there are. So for example, for many of these commands like the scatter command or the savefig command, there are a lot of options that are usually impossible to remember, but it's easy to Google it and find out.
So also like the one labeled the X and Y axis, like Google that.
Yeah. Find out. So I don't think I've labeled any of these axes, but yeah, that's something you can look at how to do. Yeah, so that's okay. So then the next step that we did was to make a histogram, and for that there is also a simple command. So what we did was to make 3 different histograms, and the number of bins is, so we loop through the number of bins. So that's the number of bins that we have in the range from -1 to 1. So the bin size, for example, for 20 would be 0.1. So again here, now, so we have this command to make a histogram. So it's very straightforward. We give it our values, the y, that's our 20 measurements, and then we tell it how many bins it should be over this range that is defined here, which we define as from -1 to 1, and then we can tell it what color and what kind of histogram also. So if we run this, we get these 3 histograms with different bin sizes. Any questions on that?
So the bin sizes that we had before were, how's the 20, 40, 80? Is that the...
Yeah, so it's a
by
number. Yeah, so it's 2 divided. The range is from -1 to 1, so it's 2 divided by that. So if we have 2 divided by 20, for example, we have 0.1 is the bin size.
What is the LW?
So that's the line width. So that's the width of the line here.
And how thick it is?
Sorry?
Like how thick it is?
Yeah. So you can make it either thicker or thinner with that command. And again, also this hist function has many more options than I've shown here. And again, just to look it up online if you want to control more different things. So after the histograms, we looked at the cumulative distribution. So now, again, remember what we did for the cumulative distribution. Now we first want to sort the values, and then that will be our x-axis, the sorted values. And then we want to take 1 step in the y-axis every time we find a y value. And we want to go from 0 to 1, and points again is 20 here. So we want to take 20 steps. And apart from this transformation to get the cumulative distribution, we just now use a command called plot, which is instead of, before we use scatter to make points, like in a scatter plot, but now plot just draws a line between the different points. So now we get this. We start, because they're sorted, we start at 0, and then every time we encounter a value, we take a step up, and then when we pass through all the points, we get to 1. So then the next one we looked at was the kernel density estimate. So as you see now, the structure of these, when you plot things, is very similar. That you define your figure and your axis, and then you do either we've done plot, we've done scatter or a histogram. And then we can set the limits. We can also, of the x-axis, y-axis, we can add labels on the whole graph. We can add x and y-axis labels, which we haven't done, but that you can easily find out how to do. And then so the only thing that's new here is that we use this in the stats library. We use this Gaussian kernel density estimate function that takes our 20 points as input and then creates a kernel density estimate. And then we just plot that. So that's also very straightforward to do. And then we get our smooth curve that describes our data. So now, if we're going to compare measurements. So now they've measured 5 different things. So it's 1, 2, 3, 4, 5. And now we show them in different colors. And in this case, we have made 20 measurements for each point. And here we've made 200 measurements. So what we can see here is that if we plot all the data points like this, we get both reasonably good first approximation if these different measurements are different, and we can also see how much data there is to support it. So in general, if you don't make too many measurements, try to show all the data. You can show, when we're going to later talk about how you can show some summary measures like mean, median, and variance also. But even then, try to overlay all the data if you don't have too many points. It's far too usual that people just show the mean and the standard deviation, for example. And that doesn't give people, you can't judge what the quality is as well. So it's definitely, if you can show the data, that's good. So now this just shows, in the case for these 20 measurements, so now we show both the histogram here and kernel density estimate of this data, the number 1. And for the case of 20, these two, and then for the case of 200, here and here. So now we can lay on the other ones. So this is now the second one, which is, we can see that they look like very similar. So this is comparing this data set and that data set. And now we're going to add on this yellow one. And we see that this one is, at least for when we have 200 points, it looks like it's slightly off. It's difficult to say for the 20 points, at least for the histogram. And here is where we see that why it's an advantage to use a kernel density at the estimate, because if you compare these two, I wouldn't be able to say whether there is any difference here, but here it looks like the two first ones are very similar and the third one is slightly off. And then, of course, this data point is far off and we can see it very clearly. And then finally, this last one is somewhere in between. So we can do this with comparison with histograms, with kernel density estimates, and we can also do the same thing with the cumulative distribution. That also, in this case, work pretty well. So this is the first measurement, and then the second one is pretty much indistinguishable. The third one is slightly off, and you see that the cumulative distribution picks up this small difference pretty well, while the histogram didn't as well. And then, of course, the green one is far off, and finally the purple one is somewhere in between. But definitely, this, even for the fewer data sets, data points, the cumulative distribution picks it up nicely.
With these smaller data sets, I mean, obviously you're showing it's the same with the larger, smaller data sets, but the kernel density distribution, you were saying you can apply a Gaussian distribution to each data point? Is that
Yes, that's what, or some other distribution. You can choose any distribution, but
right.
we chose the Gaussian.
So then when you're doing that, you're, you know, how could you say, is the line distribution you have on the next slide more appropriate because you're not normalizing the data at all? You're still, I mean, you are in the fact that you're making a relative frequency.
Yeah, you mean that here we don't make any model assumptions
right.
here. Yeah, that's definitely, yeah.
But it would require here, like before you had the, where you were looking for the systematic error and things like that, if you had that systematic error, would a distribution like this, that would all kind of, you'd have to normalize at that point for something like that or fix that, wouldn't you? Compared to when you're starting to look.
Yeah, so I mean, the visualization part, of course, later on, you're going to learn how to apply proper statistical methods to look at differences and decide whether something is different or not. So this would be something you do just before, just to get a feeling for your data, and look for systematic errors, see if there is any difference at all. So just to get a first idea of what your data is about. And then often you have many dimensions. This is a simple example that we only measure one thing, but nowadays people measure lots and lots of things. So then but it's still good to try to visualize your data and to get some feeling for that nothing crazy happened during the measurement.
Right. So I guess my main point was something like this, we are not making any normalization. Like this simply is looking at your data the way as it is, as opposed to the normalization.
Yes, yeah. So definitely that's why this is it's a simple and very robust method, because we don't make any assumptions. So now we're going to look at 5 data sets again, but these are slightly different. It's probably easiest to see in the case where we have 200 data points, of course. You see, this is the same as the number 1 from last time. So it's a normally distributed data. But then if we look at this second one, we see that there is this set of points is slightly higher. So now, again, these we plotted in the order that they were acquired, so we can to be able to see these kind of systematic changes. So it seems like something happened here towards the end, and then here we see that something seemed to have happened in the middle, and these are more continuous changes during. So all these 4 data sets contain some kind of systematic errors that we can see from just plotting it, doing scatter plots in the order that we got the measurements, or according to some other parameter. But then if we look at, now we're just doing the kernel density estimate and the cumulative distribution. So now this is of the first one. Then we look at the second one, and here it's not so, it's much clearer when we look at it here, but even in the kernel density estimate for the larger number, we see this bump, which corresponds to this systematic shift at some point. And then, but for the cumulative distribution, we don't see. I mean, there is some deviation, but we really can't tell what that is about. In the case of this, where we have 2 groups, really, we can see that we get a double peak, even in the case when we have few data points. And then we have this, where there is a continuous shift. We see that it's shifted, but we can't really see very much from these distributions. So it's much clearer from these scatter plots what's when we have a systematic problem. So then we're going to look at the code to generate some of these plots also. So to generate those first 5 sets of data, we started with having a loop. So we generated 5 different samples, all of size 20, from this, again, from the same random normal distribution, with the standard deviation of 0.1 and the mean of 0. But then for the second one, so the first 2 ones we don't change, but the third one we add 0.1 to change the mean to 0.1. Then the third one we change the mean to 0.5, and the fourth one we change to 0.3. So what we so that's too much to print, so I didn't actually show it here. But you can print it out and see what happens. But then if we now plot this, and so what we did was we also plotted these in different colors. So now we have defined this vector that we're going to use that has the RGB codes for 6 different colors that we used. But otherwise, the same thing. We do the same thing. We define a figure and an axis. Then we loop through the 5 data points, and for each of them we create a scatter plot, and then we save the figure. And then this is what the figure looked like when I generated it. So what you see is that the first data set we plot around the x values 1 with 1 minus 0.2 and 1 plus 0.2. So that's this range here. And then we choose the one of the different color for each of them.
David, I'm sorry, can you say again how you were assigning colors to each one?
So we have this array of 6 colors, and then we loop through with this variable j, starting at 0 the first time, and then 1, 2, 3, 4 it goes through the loop. And then we say that color is equal to so we use j here. So when j is 0, we're going to pick, it's going to be 5. So we're going to start, when j is 0, we're going to pick this last color. When j is 1, we're going to pick the next to last, and so on.
Is there a reason why you did this order? Or you just go up from J and increase J?
You could have, then it would have been just opposite, yeah. I think it chose it because to start with the darkest color, maybe, or something like that. I'm not sure. Okay, so then sometimes we measure two variables and we want to look at the correlation between them. And we're going to look at statistical methods later on how to correlate things. But one simple way is to, again, just visually look at it. So in this case, we have a few data points and then more and more data points for the same, the correlation between these two variables. So we plot one variable on the x-axis, another one on the y-axis. So now what we've done is we've every point here is not transparent, so that if we put it there, so we don't get when we have many data points, we don't get a good feeling for what happens inside of here. So one thing that one can do if one has a lot of data points is to make them transparent, so that it will look like this instead. So now we're looking at the same data but with different levels of transparency. So here we don't have any transparency in the point, and here we have more and more. And as you can see, we can get much more an idea of the structure when we have more transparency. And of course, the choice of how we choose the transparency in this case is depends on how our data, how the data density is.
So is the transparency then almost like a heat map, where you'd have more?
So this is not a heat map. We're going to look at the heat map.
But how does transparency work? Like how do you?
So
how does it break it down?
So in the scatter, so there is you can set the transparency. It's called alpha. So you can set it to, so alpha equal to 1 would be non-transparent, and then you just, because this is just a scatter plot so far. It's not, so here we've set, probably alpha equal to 0.1 or something like that. So it's very transparent. But we're going to look at a heat map also.
Yeah, no, I just meant more of like how does it actually calculate this transparency? Like I understand that there's a range, but just like what that alpha level is doing. And my thought was, if this is a scatter plot, maybe it's if there's points that are overlaying on top of each other.
Yeah, exactly. So that's
what it's providing as dark.
Yeah.
Whereas if you have a few.
But you have to choose it. So you have to choose what transparency you want. So that's, again, the best thing is to experiment with that and to decide what makes sense for your data, so that it becomes as clear as possible. So now so this is, again, our original plot, and then when we added the transparency, but we can also do a two-dimensional histogram or a heat map. So in this case, we have the color scheme here is the how many points are within each pixel here. So then, of course, this also gives us more information, and one can see sometimes just adding transparency is the better choice. Sometimes doing a heat map is the better choice. And that's difficult to know in advance when to use which one. Then one can also show the same data in a three-dimensional histogram. And that's usually not useful. It can look nice, but there are very few cases when that's actually useful. So just to look at one case where it could be useful to do this, if we have our original where we don't add any transparency, it looks like there is one big distribution. But as we add more transparency, we see that there actually seems to be two distributions that are overlaid here, and there is some more structure to this. And if we look at this in a heat map, we also can see it very clearly that there is another peak here. And in this, if we do the two-dimensional one, the three-dimensional histogram, then we can see that if we turn it the right way, we can also see the same thing. But again, to me at least, it's much clearer in this case to use the heat map than to try to show this histogram. A lot of people have written a lot about visualization, and there's actually one very good blog on Nature Methods that has usually very short articles that about some aspect of visualization that's worth having a look at. In general, it's not easy to make really good visualization, and one has to spend time on it. And some people it comes naturally to, but most people have to just think about it a lot and spend time on it. And this is a very good place to start, I think. So how am I doing on time? Okay, good. Now moving on to the descriptive statistics. So first, what we usually do in statistics is that we are interested in a population. So we take a random sample of this, and then we calculate some statistics on that random sample. And the first step is to do the descriptive statistics that just describes the sample. And that's what we're going to do today. But then in 5 lectures, you're going to learn about how to make then inferences on a whole population. But that's not the subject today. So again, we're going to look at a few different distributions. So we've looked at the normal distribution. So now we have our normal distribution, same one as we had before. We're going to be taking 5 different samples. So here the sample size is 3, 10, and 100. And then we just plot the distributions. So you see, if we just take 3 samples, we have a huge variation. So this time the points were very close to each other, but in this third sampling they had quite a spread. When we have 10, it starts to stabilize a little bit, and at 100 we get definitely a much more stable. So this is our normal distribution. Another one we're going to look at is a skewed distribution. So here we see that we have on one side a much longer tail. And we can see that for example here, that it's usually quite dense in one end, and then there are outliers. So we see this. We can see this tailing in the data when we have a few more data points. The third distribution is one that has very long tails. So we see here that we have a lot of outliers. Much more if we compare it to the normal case or even to the skewed case. There is many more outliers because of these long tails. And finally, we have a very complex distribution that has several peaks, but we can still sample from it. But you see that here now we get a quite a different distribution than in the other cases. So we're going to follow these distributions and see what happens when we have different sample sizes and we calculate means, standard deviations, and medians and interquartile ranges. So first, the mean. You all know that you can get the mean of a set of samples by adding the values up and dividing by the number of samples. So that's straightforward. And then, so if we look at the normal distribution and we do different size samples, we get that the mean varies a lot when we take few samples, but then the variation becomes smaller. And this is the case for every. That's why when you do measurements in the lab, you should try to repeat them as many times as possible. That's of course problematic because it might cost a lot of money and take a lot of time, but you will definitely get much better results and if you do a few replicates, and preferably as many as possible. So now, if we look at the same thing, so this is the same as before. If we sample from a normal distribution, we see that this settles down and we get good measurements as we increase the sample size. Then if we have a skewed distribution, the situation is not as well behaved, but still we have more outliers, but it's still pretty reasonable. In the case of when we have a distribution with very long tails, of course, then we don't get as good of a convergence at all. And in the case of a complex distribution like this, it's all over the place, at least when we zoom in. And one can also discuss if the mean has any meaning at all for a complex distribution like that. And it might or might not, but one should definitely think about that. So in most cases, when we measure something, we hope for that we have this case of the normal distribution, because then we can make a lot of statistical assumptions that are not always valid for these other types of distributions. So then we can also calculate other things. So we can calculate the mode, which is the most common value, the maximum, the minimum, and there are cases where we need to look at those. We can also calculate the median and different quartiles. So the median is that where 50% of the sample is below the median and 50% above. We can calculate that for any percentile we choose. So if we compare the mean and the median, so for a normal distribution, they're going to be identical. So it might be a little bit difficult to see here, but the median is in gray and the mean is in purple. But there is some variation, but it's very similar. So, but for the skewed distribution, it's not the same, and we see that the mean is generally above the median. And that's not surprising, and that's what we would expect. Then we have the distribution with the long tails. And then what we can see is that the median is a more robust measure, and that will that the spread in the mean is much larger, at least than the spread in the median. And then the same for the complex distribution, we get that we just have a very large spread in both. So these so far were measures of where the middle of the distribution is, pretty much. So the mean and the median, we're trying to tell where the distribution is. But so now we're also interested in how wide the distribution is. And that's where we get the variance. The variance, or which is the square of the standard deviation, is probably the most common measure that we use. So if we start with that, we see that for the normal distribution, we get at small sample sizes we have some spread again, same as for the mean, but then it settles down and slowly decreases. For the skew distribution, we have a larger variance, and for the long tail distribution, we have a much larger distribution. And again, for this complex distribution, we have the largest variation. So now,
Hey, David?
yeah.
So does the variance speak to the range of the values? Or
Yeah, so it's sort of
how far they are away from?
Yeah, so it's sort of an estimate of how wide the distribution is. Now, it's not a robust measure of that, because especially what we see in the long distribution with the long tails, for example, is that at random we're going to get values out here quite often that will skew. That will make the variance much larger. So it's so the variance or the standard deviation really works best for the normal distribution, because that's sort of how the normal distribution is defined. It's if you have other types of distribution, it's probably better not to use the variance or the standard deviation. And that's when the interquartile range can work much better, because it's much more robust. So what it is that you take the middle 50% of the values and you look at how big the spread is of them. So how far the middle 50% spreads out. So you don't worry about 25% on each side. And you can, of course, that's a convention that people have come up with, that they use this. You could also take the middle 90% if you want to. But that's up to you to try which one works best. So now for the normal distribution, the interquartile range and the standard deviation is related, and one can calculate what the ratio is. So what I've done here is to scale the interquartile range by dividing it by 1.349, which is then should put it on the same scale as the standard deviation. And then we see that the gray and the purple overlap quite well. So there is, for the standard deviation, there's no, for the normal distribution, there's no point. You can do equally well either of those. So for the skew distribution, we're going to get slight difference, but not a very large difference between them. But for the long-tailed one, you see that the gray and the red separate. And we get a much lower spread in the interquartile range than in the standard deviation. And then for the complex distribution, we also get some differences. So now, luckily, for a lot of distributions, we have the central limit theorem applies. So if we take a sum of values, usually a large number of values, the question is what is large, and then we'll look at that a little bit. So if we have many distributions converge to normal, so if we so sums, or we can take averages then, which is the same as the sum, then we can treat them as normal, which is very useful that you'll see in several lectures later on. So what needs to be fulfilled is that the values are drawn independently, and they are from one distribution, and they have a finite mean and variance. And for a lot of cases where you will do measurements, these criteria will be true. So that's something to remember. So what it looks like is that, so now for the normal case, of course, we already have a normal distribution. So if you take averages of 3, 10, 100, they will be normal also, these distributions. But that's not a surprise because we started out with a normal distribution. But what you can see here is that the width of this distribution becomes thinner and thinner. So we can define the mean better and better. So again, if we start out with a skewed distribution, the mean will, for a few points, still be skewed. But then pretty soon, as we go up in sample size, we'll have something that gets closer and closer to normal distribution. And then also for our long tail distribution, we see the same thing. And even for this complex distribution with many peaks, we do need more larger sample sizes to do that. But again, if we at large enough sample sizes, even for such a strange distribution, we get in the end something that's pretty normal. So this is the central limit theorem, and it's very useful because a lot of the statistical methods that have been worked out apply to normal distributions. So as you saw here, in addition to getting more and more normal, the distribution of the means also get narrower. And they actually get, so the distribution there, so what we can look at is the standard error of the mean, which is the sort of this, defined by the width of those distributions. So the standard error of the mean is related to the standard deviation of our distribution, but we divide by the square root of the sample size. So that's the, if we collect larger and larger samples, we can calculate the mean more and more accurately, as we see from these distributions getting more and more narrow. So then the third exercise, so if you have this script to look at, so here now we're going to first generate the skewed data set. And the way that we're generating this is by random by sampling from the distribution. So this method can be used to sample from any distribution. In general, so the first step is to generate a pair of random numbers within the range. So these are this x underscore test and y underscore test. So now we use a uniform distribution in both x and y, and the range is, the y-axis range is from 0 to 1, and the x-axis range is from -1 to 1. And here we need to, if we want a sample size of 1, we need to generate a few more. In this case, just to be on the safe side, generate a lot more, 30 times more, because we're going to reject a lot of them. The way we're going to do it is, if we have a distribution, now this is actually an experimentally obtained distribution that we want to resample. So what we do is we generate a random x value and a random y value. And then we see, is it inside of the distribution or outside? And if it's inside, we keep it. And then we again generate another x and y value. Now this time this is outside, so we throw it away. And then generate another one, it's inside, so we keep it. Another one, it's outside, throw it away. It's inside, we keep it, and so on. So we continue this, and if we randomly sample with uniform distributions over the whole x and y range, we're going to resample the distribution. And this is a very convenient method to use for any distribution.
I just want to say, when I put this in, it's telling me that skew is undefined.
Oh, so yeah, because that's, sorry, I didn't show that here. That's in this, if you run the script, it's defined in the script. If you, did you download it? So if you run the script, it should be defined there. Yeah, I didn't want to show it here because it doesn't really matter how skew is defined, because that can be defined in many different ways. The point was just to have any skewed skewed distribution. So this code, this is what we do here, is that then we have these, the two random x and y, uniform distributed. Then we calculate this y test 2 from the skew function, and we just normalize it so that it's between 0 and 1, and then we see if it's smaller. So this is the test whether it's inside the distribution or not. And then we just limit it to our sample size. So that's how this general procedure goes. So then we can, so now we have a skewed random data set. Yeah.
What if you're, say, working with a data set where you don't have this limit, so you don't know, say you just, like you're looking at a data, like a database, and then you have like a lot of data, and you don't really know where the range is. Like, how do you know this is not going to just come on forever? Like, keep going outside or in and out.
Yeah, so that's, I mean, usually you can define your x and y limits. And I mean, for this method to work, you have to make some decision on what your range of interest is. The biggest problem with this method is that if you have a distribution, like, let's go back to the, so if you see here, this is what we're sampling from. So most of the time, anytime a point lands outside of the curve here, which is difficult, but I mean it's probably going to happen at least 80% of the time here. Those we throw away. So it's not a very efficient method to do this, and there are many much more efficient. The advantage of this method is that it's easy to implement and it works for any distribution. But it's so you can have an even worse case where your distribution is a very small part of the area, so that you're just wasting a lot of computational time. That can definitely happen. Oh yeah. So then in the script, so now there's a lot missing here. So don't try to enter this into the... but in the script, what it does is that now here is where it generates a sample. With these, that's replaced with these 3 dots. So it repeats that 1,000 times, and then calculates the standard deviation of this, the averages, and then it plots the histogram of all the averages. And it does this for the, and it adds here a title, saying what the sample size is and what the standard error of the mean is. So here is where we have 3 cases. We have a sample size of 3, sample size of 10, and of 100, and it calculates what the standard error of the mean is in this case. So again, if you run the whole script, you should get these outputs. Okay, so then we're going to talk a little bit about box plots, because they are, we have time. Because they provide a quick overview of the essentials of the data. And the basic box plot is that we have a box between that show where the 50% of the data is in the middle. So between Q1 and Q3. It also shows the mean, and then it puts these whiskers at 1.5 times the interquartile range. And then anything that outside of that, it puts the outlier, the actual data points as outliers. And then you can have a more complex one, but we're not going to talk about that, where you put a notch to set the confidence interval, show what the confidence interval is. But you're going to learn about confidence intervals in later talks. So now we're going to do the same thing. We again have our 4 different distributions, and we're going to now present the data as box plots instead of the individual values. So now again we have different sample sizes, 5, 10, 100, and we do 5 different samplings. So we see that when we have few data points that we select, the boxes vary a lot. But when we have more data points, the size of them stabilize. And in the case of the normal, if it's a normal distribution, we actually end up having very few outliers, even in the case when we have 100 samples. For when a skewed one, we see a similar trend, but what we see is that we have more outliers, and of course they're in the direction of where the skewness is, where the long tail is, as you can see here. So now, if we have a long tail distributions, we have a lot of outliers. And for the complex distribution, it gets it's just not a good way to present the data. So now we can choose to do this. We can also choose to show all the data points. And that's what I would recommend in a lot of cases, that you show a box plot and you show all your data. Because then one can see if is this box plot based on just a few values or on many values. And that can help the reader quite a lot to see actually how, what the box plot is based on. So now, if we just, I'm almost done here, but we're just going to compare now a few different ways to show the same data. So a quite common way to do it is to show error bars, show a bar graph with error bars. So now what we see is that we have a few measurements. There seems to be a trend here. Probably these three values, at least, we can say are higher than the others. And then, so when we show error bars here, we can either use the standard deviation or we can use standard error. Or we can use the confidence interval. And there is different people use different things, and it's at least one should very clearly say what error bars were used, because otherwise it can lead to quite a bit of confusion. Another way that would probably pretty much show the same thing, maybe slightly better, is to just show the mean value and the standard deviation. So the advantage here is at least you see the error bars in both directions. And if you have asymmetrical error bars, that could be easier to see this way. But if we compare, yeah.
Sorry, what was the difference between standard deviation and standard error?
Sorry?
What was the difference between standard deviation?
Oh, so the standard deviation is what we
plus minus.
calculated before, when it's related to the width of the distribution.
Yes.
The standard error we divide to get we can get the standard error from the standard deviation by dividing by the square root of the number of samples we have. So that's why they are smaller. But then now, if we instead show box plots, we get I think much more information in a compact way. So definitely, it would be great if you learned how to generate these box plots so you can show your data this way. And here we only show outliers, but in this case we show also show all the data points. And so, for example, what we didn't see from any other of the plots is that we actually had very few data points here and much more data points here. So in a case like this, it's definitely recommended that one shows that because then otherwise people will not know. From this, one would definitely not be able to tell that that's the case. So then same thing, but for the skew distribution. Just to bring home the point that if you show box plot with all the data points, that's definitely preferable. Even if you have quite a few. It's probably 200 data points that are shown here, but you still get a pretty good idea of what's going on.
And what I've learned before, and maybe as you're suggesting this with normal distributions as well, it might be different in your opinion, but to me it's always been taught that if you have normally distributed data, that using standard standard deviation is usually appropriate. So if you're looking at a clinical paper, you would be seeing the middle section there if it was normally distributed. If you're looking at interquartile ranges, or looking at the variance in that way, that's normally indicative of a non-normally distributed data set.
Yeah.
So you'd be using parametric versus non-parametric statistics. Is that convention normally held up in what you guys do, or how you teach it, or would you suggest almost always to go to a interquartile range box plot scenario to show your data?
Yes, I think that's the box plot is what we recommend, and because it just gives you more, and a lot of times we have too few data points really, so whether it's normally distributed we can't often tell very well because we have so few data points. So that's why it's probably, when presenting it, it's probably best to not make too many assumptions on that. At least when visualizing it. I mean, one can always then, when applying statistical methods, if you describe it properly, what you're doing, then you can definitely make assumptions. Because I mean, you're going to get much more power if you do make normal assumptions. But when showing the data, it's best to show all the data and using box plots, I would say. So now, exercise 4 is to do to plot box plots. So again, here now it's pretty simple. If we have our array, which is called examples here, we can simply call this command box plot, and it will create a box plot. And after we've made the box plot, we can change the colors of the boxes, the whiskers, where the median is, the caps, and the flyer. So all these things we can change, and so the flyers, these are the outliers. So we can control all of those things as we want. So in this case, it would be just the output of this script, which were again 5 data sets, and each of them have a box plot and showing all the data. So just to summarize this part, again, what we looked at here was descriptive statistics. So we take a sample from a distribution, and we looked at a few different example distributions that had different properties, and then we calculated descriptive statistics that characterizes that sample that we took. So we calculated means, medians, quartile, percentiles, to sort of get an idea of where the distribution is on our scale. And then we calculated the variance and interquartile range to decide what the spread was of the distribution. And then finally we looked a little bit at box plots and other types of graphs, but definitely use box plots if you can. And also, Nature Methods also has a short articles on statistics, and every month there is a 2-page article on some aspect of statistics that I think would be very good complement to this class to read, and you can go and on the website that's indicated here. So the homework for I guess it's due on Sunday, would be to take this script, the exercise 3 script, and modify it. So this exercise 3 script was the one that looked at what happened if you sampled sampled many times and calculated the average and looked at how the distribution changes. So what we want to look at here is the relationship between the standard error of the mean and the standard deviation. So the idea is to take the ratio of those two and see how that ratio changes as a function of sample size. And we looked at that, and it should change as 1 over the square root of the sample size. But you should just change the script and show that you can get something that's close to 1 over the square root of that. When you're done the homework, you should just email it to the regular place, both the script and the plot. And so if you need any help on Thursday, there will be tutors available. And also, I mean, anytime if you have any problems, you should definitely come after class on Thursday, but you can also contact the tutors to if you have other questions or you want to meet with them separately. They're available and they're there for you to use them if you run into problems. And next time, Stuart is going to talk about sequence alignment.